So if you want to follow along with me, you can download the tiff here at www.suite2p.org/testdata/gt1tiff. So this is a tiff I'm going to run during the demo. Okay, so let's get started. So in calcium imaging, there's been an explosion of new techniques. The example here is dual plane imaging, where you can image two planes simultaneously. In this case, in layer 2, 3, and layer 4 in visual cortex. And so we need, with these new imaging techniques, we need fast and accurate pipelines to process this kind of data. So this is where something like suite2p comes in. So we're able to take these types of large scale recordings and extract many thousands, in this case, around 50,000 neurons from these large scale recordings. And so what does suite2p do? How do we get to this point where we have all these neurons? So a brief outline is we first do motion registration. So many of you are probably recording from awake and behaving animals. So their brain is going to jitter around while you're recording. And so first you need to do motion registration to make sure the frames are steady. And then you're going to detect, and then what suite2p does is it detects cells in these recordings. And then it extracts the signals from these cells and performs spike deconvolution. And then finally, there's a quality control step that's optional for the user, where you can go into the GUI and you can decide which ones are cells or not cells. And you can use our built-in classifier or train your own classifier to classify cells. And so all of this processing should take around an hour or less on a standard desktop computer. And so ideally, what suite2p we really want to enable is this kind of that every day you can process your data overnight or the same day, and you can analyze your data so that you can modify your experiments day by day as you see potentially changes in neural activity and so on. All right, so how do you get started with suite2p? So if you've never used Python before, you need to install Anaconda for Python 3 for your specific operating system. And then you're going to need to download this environment.yml file. So we're going to create a Conda environment for suite2p. So if I want to do that, I'm going to go to, here I'm at the GitHub for suite2p. I'm going to go down, I've got the same instructions here too. I'm going to take this environment.yml file. I'm going to copy paste the text from it. You could also download the GitHub repo and get the text. I'm going to copy it into this notepad here, which I've already saved as the environment.yml file for myself. And then the next step is I'm going to create the environment with conda m create f environment.yml. So here I've got my, in this case, I'm opening an Anaconda prompt, but if you're on Linux or Mac, you would be opening a terminal. And so now I'm going to type conda m create f environment.yml. And it's going to start creating this suite2p environment for me. And then after it's created, we're going to activate conda activate suite2p. It will tell you to as well. And every time you want to run suite2p, you're going to have to do this conda activate suite2p step. And then to run suite2p, we'll do Python. You do Python m suite2p, running the module suite2p. And also, as with a lot of software, NWB also updates, and suite2p does too. So you might have upgrades that you need to upgrade. So if you want to upgrade suite2p, then you would do pip install suite2p upgrade in this conda environment. So it's running right now. It's creating the environment. So once you create the environment, you can open suite2p with this Python m suite2p command, and it will open a GUI window that looks like this. And then you'll have this, there's all these different file menus, and we're going to, in this case, we're going to run suite2p. But there's also this new addition, which is this load NWB file. So you can take an NWB file processed from any pipeline and load it into the suite2p GUI now. But right now, what we're going to do is we're going to click on Run suite2p, and it's going to open up this window so that you can run suite2p in this GUI interface. And so you can add a directory with tiffs here. You can click Save NWB as 1 because we want to get an NWB output. And then something else you could set to 1 is this Keep Movie Raw. And this is going to let us visualize the original movie along with the motion register movie, but that's just optional. So let's see, this worked. You can do the same thing here. Can everyone see things okay?
Yeah, it looks good.
Cool. All Also, after, like, feel free to stop me with questions at any time. I'll naturally pause after like each module to ask for questions, but feel free to stop me at any time too. It's going to be hard for me to monitor the chat as I'm going through this, so feel free to unmute yourself and say hi. Okay, so starting up suite2p. And this will take a second. And now I'm going to do what we were saying, run suite2p. My computer is getting a little slow with Zoom, I think. I'm going to shut this off too. Okay, I think my computer might be too slowed down by Zoom, which is unfortunate. So yeah, okay, we'll just keep going. Sorry, I have not done this demo over Zoom before. I've only done it in a classroom. So yeah. All right, so we'll go through, we'll see if this ends up working, but I guess my computer is, yeah, this is unfortunate. Yeah, all right, I'm sorry, everybody. All right, I'm gonna keep going. I think my computer is just consuming too much with Zoom, I'm not sure. All right, so let's go through the different steps of suite2p. So first we have the motion correction step. So this is where we have a movie. We have this movie of our data, and this is jiggling around because the mouse is moving. In this case, we're recording from mice. And so we want to correct this movie so that there's no motion in the frames. And so what we do in suite2p is we find a reference image, which is going to be what we align all of our frames to. And we have a way that we do this in suite2p that I won't go into, but hopefully you have a nice crisp reference image, and then we're going to align each frame to this reference image so that all these frames are on top of each other, basically. And so if we look at this example frame here, you can see, if you look at the difference of these two frames, you can see that it's shifted over to the right from this frame. And so we want to figure out what the shift is between these two frames. And to do this, and to have very accurate shift detection, we first whiten these images. So instead of using the raw images, we whiten the images, and then you get these crisp outlines here. And then you can align these outlines. When you have these edges, you can get a much crisper correlation between these two frames. It'll be much more localized in space. And so I'll show you what I mean by that. So you take this frame, and we're going to run it across every possible pixel shift on the image, on the target image, the reference image. So we move it across, and that's going to create this matrix, this correlation map here, which in this case on the whitened images has this sharp peak that's specifically at this location. But if I didn't whiten my images first, actually you get something that looks more like this, more of a blob. So we're able to get better accuracy when we do this phase correlation step first. But yeah, you can also turn off this step if you want in suite2p, but I wouldn't recommend it. Okay, so this will work if you just have motion in the whole frame, but there could also be motion which is different from different parts of the image. They could move differently. So you need to use a non-rigid registration in addition to this in order to register them. So what you do for non-rigid registration is you divide the field of view into blocks now. And now we're going to do this phase correlation on these blocks. And I'm going to try to restart this again. And hope for the best. Okay, so we divide this field of view into different blocks, and we're going to take the phase correlation for each of these blocks. And so this will give us those little maps I was showing you. We'll get a little map for every single one of these blocks that we divided the image into. And then so each of these maps corresponds to a pixel shift. So these correspond to these arrows, which say, okay, this part of the image moves in a certain way, and then this part of the image moves in a different way. And so this is at the block level. We need the shifts at the pixel level in order to move the image around. So we take these correlations at the block level and we interpolate using bilinear interpolation and get a pixel shift for every single pixel. And then we shift all of these pixels and have our final image. And then we can also, so there's ways in suite2p that we can visualize whether or not this registration worked correctly, which I'd like to get into. But first, so does anyone have any questions about motion registration? Okay, I see the chat going. Hi, Carson, can you estimate the fractional shifts? It seems to from the, oh yeah. So we actually, we do an upsampling using kriging interpolation so that we can get subpixel shifts on each of these blocks actually. We don't do it for the rigid shifts, but we do it for the non-rigid shifts. So these correlation maps, which you can see is there's this peak here, and we could take the peak, just the top pixel, but you could also fit an interpolation to that. And in our case, we use kriging interpolation and take the peak of this interpolated value, and that will give you a subpixel shift so that you're going to have pixel shifts that are potentially more accurate because you could do a shift of 0.1 rather than 1 pixel or so on. Yeah, and so we only do that as well for the non-rigid because that's when we're actually shifting all these pixels, and so yeah, so the rigid is very fast, and then the non-rigid we have to do this whole interpolation, and it's a bit slower, but we've sped it up by implementing all of this in Numba. Oh, yay! Okay, my computer's not dying now. All right, so we're going to add the directory to the data path. So this is the directory where I downloaded this tiff that I was telling you about. I'm going to set save NWB to 1. I'm going to set keep movie raw to 1, and I'm going to run this now. And now it's running. It's converted these tiffs to binary format, which is the format that we put all of our frames into. And now it's running registration. All right, so we'll let it keep running in the background, and then I'll show you this, how to quantify the motion after. I'll tell you briefly what this is because I think this is really useful as a user, especially like when you're first getting started with your data. All data is very different. So depending on what kind of data you have, you'll have different types of motion. And so what we do in suite2p, we have this extra step after registration to test whether or not registration worked well, where we take our whole movie and we take the principal components of that movie. So we have our motion corrected movie and we take the principal components of it. And the hope is that if I take the principal components of my movie, all those principal components correspond to activity. But there's a potential, if there's motion in your movie that's left over, then if I take those principal components, actually it might find principal components that correspond to motion and not activity. So we take these principal components and we take the top frames in the top principal component and the bottom frames in the principal component and take the averages of those frames. And so these give us 2 frames which are kind of at these ends of this spectrum, and we compare these frames and say how registered to each other are they, like how similar are they to each other. We do this registration step to them, and we see like how far off these frames are from each other to see if these PCs correspond to motion or if they correspond to just activity. And so once this runs, we'll look at it in the GUI and you'll get to see what these things look like. All right. And so I'm going to go to cell detection now. Are there any other questions? And I have the chat open now, so you can chat if you don't feel like asking a question on the mic. Sorry. Okay, so how do we do cell detection in suite2p? So we have so in suite2p, we're able to detect these ROIs. Here's a zoom in of a field of view, and we can also detect many thousands of ROIs if we do a multiplane recording, for instance, or a mesoscope wide recording. And so what are different ways that you could potentially do cell detection? So one technique could be to do an anatomical cell detection. which is where you have your mean image and you find ROIs on it. And this might work okay, but if you're going to do anatomical segmentation, what will work even better is if you use a little bit of functional information. So for instance, you could take the max projection image, which is where you take the maximum value of the pixel over all of time. And this will give you a nice looking image like this. And so you could try to do cell detection on an image like this. And you could use any anatomical segmentation algorithm. For instance, if you use the one that we've developed, CellPose, you're able to find around 679 ROIs on here. So you can find a decent number of ROIs with anatomical segmentation, but you're not necessarily going to find all of them. And the ROIs you find might not necessarily have activity. And so here's a figure describing this. So in this study, what we did was we manually labeled cells using the mean image. This was me, and I'm not particularly good at labeling cells by eye. And then we also ran the suite2p algorithm on this data, and then we looked at what the activity traces were for these manually labeled cells versus these cells found with a functional segmentation algorithm such as suite2p. So Kaiman or CMMF is another example of a functional-based algorithm, and there's many other ones as well. So you could pretend like this is any of those types of algorithms in a sense. So if you compare a structural algorithm versus a functional algorithm, and you look at the skewness of these traces. So for instance, if we look at this trace here, it's very skewed. Whereas a trace down here, this is a trace with low skewness. So these high skewness traces are these traces that have these big blips of activity that correspond to these GCamp events. And so the functional ROIs, unsurprisingly, they're found by finding these big sort of events that are correlated across pixels. And so these ROIs have high skewness, whereas if you look at the structural ROIs, they actually have skewness, which is pretty similar to if we just drew random ROIs all over the image. So with a structural or anatomical method, you'll find potentially different ROIs, but not necessarily ROIs that have activity. So ideally, you want to use, if you care about finding activity, you really want to use a functional-based algorithm, and potentially you could combine these two, and that's something we're planning on doing in the near future, is combining CellPose and suite2p to find as many ROIs as possible as well for the user. All right, so how do we hunt for cells? So we had kind of this idea you could use the max projection image and use a segmentation algorithm on that. But in the functional imaging, like if we want to use a functional algorithm, we need a similar sort of map which represents the activity of the cells rather than this static image. And so we create a map that looks like this, that's based on the correlated activity of pixels. It's a multi-scale sparse projection map, or you can call it a correlation map, where we basically, for every pixel here, we find how much variance is explained of a square around that pixel of a small size, a medium size, a big size, and a really big size. And we take the maximum of that over all these different maps. We compute it at multiple scales. That's why it's multi-scale. And so then this gives us a picture that looks like this, which is the maximum over all these maps at these different scales. And then the algorithm goes in and it takes peaks in this map. And after it finds a peak, then it starts to grow the cells and now when we grow the cell, all right, so that's the map. And now if we have a cell, we start growing the cell. And to grow the cell, we now look at the activity of these pixels over time. And we say, let's take basically the frames that have the most correlated activity with this peak here from our maximum projection map. And then we take those frames and we start growing and say, is this pixel correlated across those frames? And we start growing this cell. So you kind of, the idea is like you want to use frames where the cells are active in order to create these maps. So you want to use these frames where these pixels have high activity, and also at the scale where you have at the scale where we found the maximum variance explained, basically. And so that's how you grow these cells here. So you start adding pixels based on whether or not their activity is correlated with the activity at the peak here. All right, and so then you get a picture that looks like this, and you actually find over twice as many cells using an activity-based segmentation such as this, or functional segmentation such as this from suite2p, compared to using an anatomical segmentation algorithm like self-pose or another algorithm like that. Okay, and so because we have this step where you're kind of growing the ROIs, and there's no kind of limitation on the shapes of the ROIs, you can get ROIs that look really long, and they're basically, you can model dendrites in this way. You can capture their activity like this. You can also capture axons and boutons. And so, yeah, so, and you can also, this segmentation algorithm works on different types of data too, from different species. So here's an example of data from zebrafish from SF Cybertal and Isaac Bianco. It works in different brain areas. I've been mostly showing you visual cortex. Also works in superior colliculus, for instance. This is data that Sylvia Schroeder collected. Also works in barrel cortex. This is data that Henry Daglish collected in barrel cortex. And so, yeah, so that's pretty much it on cell detection. Does anyone have any questions about cell detection? Looks like suite2p is almost done running on this data. All right, I'm not getting any questions here, so I'm gonna keep going. So spike deconvolution is the last step in suite2p. So it's technically an optional step, but it's very fast to run, so it doesn't hurt to just always run it. And what spike deconvolution helps with is it increases your temporal precision of your traces. So this is particularly important if you're, for instance, in our case, we're imaging in visual cortex and we're looking at the responses to visual stimuli. And if we don't deconvolve, then our traces will bleed into the activity that's present, that it will basically bleed into the responses for the next image. So if we deconvolve, we actually, we kind of remove this bleed through, and we actually improve our signal variance. We basically, our responses are more reliable to these visual stimuli if we deconvolve rather than not deconvolving. So I recommend looking at your deconvolved data, at least, and considering doing analyses on that. There's also a temporal baselining step that happens before deconvolution. So it generally is better to use the deconvolved data. All right, and so finally,
Carson, could I ask a question about the deconvolution?
Yeah.
So I tried the suite2p deconvolution. So you get basically a continuous signal, right? And you get this like spiky activity, but they have different amplitude as well. So if you want to basically move to like some discrete spike trains, what would you recommend to set the threshold on?
Yeah, so that's a great question. So can you set I think there's another question there. Can you find a threshold that works for every cell as well? And so I think what we've seen and what other people have seen, so actually in our review paper in Current Opinion in Neurobiology, we actually do an analysis on some ground truth spike data where they have the cell attached recordings and the calcium imaging. And what you see there is there's a pretty big variance in terms of the amount of the GCamp, the blip of GCamp that occurs with a single spike. So it's hard to say how much GCamp corresponds to a single spike. And it varies from cell to cell. So I would be very wary to call these signals to set a threshold and say this is 1 spike, this is 2 spikes, this is 3 spikes. I mean, you could even see, you could test and see, like, for instance, let's say you binarize your signal and you could see how much information you lose in terms of what you're trying to decode or something like that.
Yeah, no, sorry. I'm asking because we're trying to basically deconvolve some single spine signals and then correlate it to spike sorted data from combined extracellular recordings, right?
Cool.
So that would be like a way to say, okay, this spine is from this neuron. But so that's why we need like spike times, or we need to convolve the spike train to get some sort of calcium trace. But I guess it's the same problem.
Yeah, I see. Yeah, I think no matter what, you have this issue that if you you can try binarizing in some way and see if you're able to do the correlation, and if it works. But you might be in a case where you'd get higher correlation in general if you are able to do the matching on these signals that have this continuous distribution. You might have higher correlation actually if you don't threshold it. So yeah, I don't know what your correlation matrices look like between them, but
we're still experimenting. Right,
yeah, no, that's a hard question. Does anyone have any other questions about spike deconvolution? Oh, someone asked, what about correlation image and cell pose? Yeah, so I think suite2p is able to get all the peaks from the correlation map. I think what happens are there are cells that aren't on the correlation map or don't have weak signals on the correlation map that cell pose might find. Particularly if you're doing recordings day after day and you want to get all the cells possible and you want to look at learning across time, you might want an instance where you do get all the cells, whether or not they have like strong activity on that day or not. So it might be able to pick up these cells with lower signals. Yeah. Yeah, and yeah, so that's pretty much all that I had. Yeah, so I think these points about like this discussion we had about spike deconvolution is really important. Like it's really hard to know what a single spike is relative to the calcium signal across cells, and that can vary from cell to cell. So whenever you do these analyses, if you do decide to binarize your data or not, I wouldn't recommend it, but you can always look and say, what is my decoding accuracy if I binarize or I don't binarize, and these sorts of things. Okay. So finally, in suite2p, you can explore the data in the GUI. You can also, Nick and Chris this summer made a nice Jupyter notebook that you can use, that you can run suite2p in. So here they're setting up the ops file for you, and you set up what directory your data's in, and then you can run suite2p in the notebook, and you can also look at the output of suite2p in this notebook too. You can look at the different images that come out, the max projection, the reference frame, and so on. You can look at the ROIs found. And you can look at the different traces. And so this notebook might help you, especially like if you're getting started in Python. It can help you figure out what these different outputs of suite2p are and how to load them into a notebook and how to look at them and stuff like that. Or you don't need to, I mean, you can use Python, but you can also, let's get into the GUI. So all the processing ran, and then this last bit here is the new saving of the NWB file. And so now we've saved the suite2p output in NWB format, so you can load this NWB file in Python, you can load it in MATLAB, you can load it in whatever format you want. You can also directly save a MATLAB file from suite2p if you want with this save mat option as well. And so you don't have to use Python to view your traces. All right, so what does this data look like? So here are the different ROIs that were detected in this tiff. This is a 4,500 frame tiff. And the things I wanted to show you were just a little bit about the registration that I didn't get to show you before. So I went into the menu here, I went into the registration menu, and I clicked on view registered binary. And this is going to let us look at this movie to see how well our correction worked. And we can view the raw binary at the same time. So because I said keep movie raw equals 1, I have access to the original, I kept around the original binary, that has the raw data in it. And so on the left, we have the motion corrected version, and on the right, we have the raw data. And so we can play these two movies and see, you can see this one jiggling around a little bit, and then you can see the corrected one on the right, or on the left, I'm sorry. And you can, and when you have it paused, you can use your left, right arrow keys to go through the movie quickly. And this is a nice way to see if this looks really static, then I don't know how well this or how good this looks through Zoom, but if it looks pretty static, then you know that the motion correction probably did a pretty good job. You can also view the masks here if you want to. I'm going to turn off the raw binary, and so you can maybe zoom in on a cell you care about and say, let's go to what happened when the cell was firing. There, this is cell firing, and so you can view some of the data here. Okay. And then the other thing I was telling you about was the registration metrics. So how can you tell if your registration worked well? And so that's here. So let me go through these different panels. So I was saying this idea that we take the principal components of the whole movie. And so this is so you have a principal component that's going to be made up of, like, you've got the pixel side and then you've got the time side of it. And so the temporal activity of this PC is shown in this plot here. So this is the first PC of the activity. And this is of the whole movie, of the whole motion corrected movie. And then this is what the activity of that PC looks like across time. And what you hope is, so then we take these averages of the extremes of that principal component when the principal component is high and when the principal component is low, and that gives us two images. And we're looking at the difference of those images, of the image here. And because I've run motion correction, it's going to probably look pretty good. So if we look at this difference here, it looks like it's mostly activity based. And if we looked at the merge base, so you're not seeing, like, if it wasn't activity based, you'd see like little edges of cells, like you see like little crescent shapes around cells that look like activity. And then you can play, actually this one flips between the two PC, the top and bottom frame when you press play here. And that's kind of, if you stare at it and put your head in the right place, you can kind of try to see if there's motion there. And you can go through, you can also, you can look at these, you can also look at the metrics themselves. So this is basically taking those two frames and registering them to each other and saying, how big are these shifts? And these pixel shifts are 0.1 for the maximum non-rigid shift between these frames, and the non-rigid average shift is around 0.01. So these are pretty accurate. So the motion correction did a good job on this movie. And then we can go through the different PCs. This one was a little worse, but not much. And we can again play this thing, and we can look at these different images, and we don't see very much. But this is another way too. You could see if you have Z drift, for instance, if your movie is slowly moving in Z, this would show up in these principal components too. And another thing I forgot to show you is there's a new feature in suite2p in the view registered binary view. You can also now load a zstack tiff, and you can compute the Z position of of the binary across time inside that zstack. And then this trace here will show up. That's actually the Z position of that plane across all of time. So you can also, and then it will save the Z position for you. And this might be useful, particularly if you have a lot of Z movement in your data and you want to make sure that your Z movement isn't causing some of your signals. Then it's a good idea maybe to run this Z position computation, and then maybe correlate this Z position with some other variables that you're looking at. So yeah, okay. So yeah, just in general, it's always good to make sure you look at these things to really make sure that you have high quality data going in, because if it's not well motion corrected, then your traces are not going to be great, and also the cell detection is not going to be great. And then so after that runs, you have the ROI detection, and we can look at the ROIs on various different backgrounds here. Here it is on the correlation map. And you hope that it found most of the peaks here. There's also something I didn't mention, I mentioned briefly, which is the fact that there's a classifier being run at the end of suite2p to decide what a cell is and what isn't a cell. And this classifier you can train on your own data. If you label your data, basically I can right click and I can flip cells left and right based on whether or not I think they're good or not. And you can use this training data to train your own classifier and use this classifier on your own data to decide what you think is a cell and what's not a cell. Right now, the classifier is pretty strict. I could move a few more cells over. I can set the probability. So each of these cells has a probability of being a cell or not a cell based on our default classifier. And I could set the probability that the cell has to be above like a probability of 0.3 to be a cell now. If I set it to that, I'm going to get more cells. So these cells move over to the left. These are what I'm calling cells. And now I'm at 1800 rather than I was at 1400 before. Okay. And all right, so then I've got a few more cells here. We can look at other aspects of things in the GUI. So you can look at how skewed these traces are, for instance. We can color the traces based on skewness. We can change the color map to something that people like better, which is like Viridis or other things. We can look at different aspects of the data. Something that's fun you can look at are the correlations between neurons. So this says this cell is it looks for cells that are correlated to this cell. So if you look at this, the color bar here, you can see that it goes up to 0.73. So there's cells that are up to 0.73 correlated with that cell. And if I press Control, I can click on multiple cells at a time, and I can kind of do this game where I try to find all the cells that have correlated activity here, and kind of look at all their activity together. I can do this for different types of activity. So I can do this for in this case, this activity mode is set to the corrected trace. So this is the normal trace minus the neuropil trace. I could set it to the deconvolved data, and that will change the correlation slightly. I could even change it to the neuropil. And then what you'll see are these nice kind of spatial correlations between the data, just kind of basically that the neuropil is going to have these kind of slower spatial correlations that you can see in this color map here. So let's go back to the deconvolved. And maybe you don't want to play this game of trying to find neurons that are correlated to each other. Maybe you want an algorithm to do it for you. So what we can do is we can go into this. Let me go into the random color when I do this. I can go into this visualize menu, and I can run an embedding algorithm. In this case, we're running raster map here, but you could run, this would be similar to the output of like TSNE, for instance. So what I'm going to do is I'm going to embed my neurons into a 1D space. And so this is the output here. So this is what the activity looks like of these neurons embedded. So I have 1800 neurons here. And now these neurons are embedded so that neurons that are correlated to each other are put close to each other in this 1D space, in this raster here. So this is neurons by time here. And so then I can move around and be like, okay, maybe I want to look at these neurons have similar activity. And so this red box here, what I'm doing, this red box is defining the neurons I want to look at. I can click this show selected cells in GUI. And then these cells are selected for me. And I can actually look at kind of what, oh, sorry. You can double click anywhere to get the GUI back to the right place, because sometimes it resizes somewhat randomly. You can look at where these cells are that were correlated to each other in this raster map. And I can make this menu bigger, and again, double click so that I still have that window, and you can look at the activity and how correlated it is between these traces, and looking at where they are in the map. Yeah, and that's, I mean, so the nice thing now that we have this NWB format, so I'll show you now, this actually loaded automatically from the GUI. It automatically loads the MPY files once suite2p finishes running, but I can also load the NWB file that I processed. So in this folder GT1, where that tiff was that I told you about, that test data tiff on the website, it made a folder called suite2p, and inside that folder, there's this plain zero folder which has all the MPY files, but also there's this NWB file, which contains all the information over potentially, if you have more than one plane, it will have the information for all the planes there. So we can load this in, and you can load the NWB file now. It's loading into the GUI, and you can look at the NWB file as well. And so what this means is that you can use the suite2p GUI with any, in theory, with any other algorithm. So you could run other calcium imaging algorithms and then load the activity into suite2p. And I'm gonna, okay. And let's see, is there anything else? Load custom view. I think I've pretty much shown you all the things. You can do some other things too. I can select the top maybe 20 ROIs that are the most skewed and look at them here if I want to, stuff like that. I can also draw a selection of ROIs, so I can look at the activity of all the ROIs in a single location. Yeah, that's pretty much it. Does anyone have any other questions? There's a new window now too. You can auto suggest merges. Do you use any specific ontology for NWB? What do you mean by that? I used what I learned in the first workshop. Oh, the names of the fields. Yeah, I sort of made my own names for some of the fields, which were required. So if we go to the save NWB, there's a few fields that I named here. So I made these backgrounds, and I made them mean image, vcore is the correlation map, and then max proj, and then the mean image, potentially you have it for channel 2 as well. So I made these different fields here that get saved as background images. But then I think all the other fields, like I mean, I think there were examples with the fluorescence and the neuropil as well already. I'm not sure if this is a new thing, but I think that was there before. Ben can correct me if I'm wrong. Yeah, I also made this one too. This is cell. So that's this classifier field. I think, yeah, so right now it requires the is cell field as well. And in the future, I could make it so that you don't need to have the is cell, is that it could just say, if I load it from another algorithm, just assume everything is a cell. Yeah, because I'm guessing most people don't have that is cell field. I'm glad you think it's a useful field. Yeah, cool.
Yeah, I think we started to do this in the last hackathon, but I think it would be really great if the different calcium imaging tool builders could kind of come to a common agreement about these names, because NWB does provide a lot of flexibility in how you name these things, and you certainly chose reasonable names, but there, you know, I know Cayman also chose a different set of reasonable names. And what I think would be really awesome is if you could save Cayman data and then load it into this GUI and explore it alongside having run the suite2p algorithm, right? And I think, you know, it's really the goal of NWB to provide the ability to be that common language between these different processing systems. And that last critical step would be just kind of coming to common agreements about how to name things. But yeah, this is really great. This is like such a powerful GUI. So really awesome work. It's great to see this as part of this ecosystem. Thanks.
Yeah, thanks for the help less hackathon. And yeah, I agree. Like I am happy to change. All right, cool. They have the is self field.
Oh, perfect. Okay.
So yeah, I guess most of what we have to agree then is probably on maybe these backgrounds too, and how we declare these things, because these are useful too for the user to see in the GUI for sure.
Yeah.
Cool. I will stop my share.